Hello everyone, welcome to We Learn and welcome back to the class Molecular Basis of Inheritances. In the last class of Molecular Basis of Inheritances, we studied about the DNA packaging. If we talk about DNA packaging, we already know what a nucleosome is. And a nucleosome is basically consists of an octamer plus DNA plus H1 protein. So this is basically an octamer which is basically consists of H2A, H2B, H3 and H4 protein, and these proteins are basically present in dimer form and it form an octamer. So 2 units of this, 2 units of this, 2 units of this, 2 units of this form an octamer, 8 units, therefore it is an octamer. Because there are 8 units, therefore it is called as octamer, or it is called a new body, or it is called an core molecule. So this is the core molecule. If we talk about DNA, our DNA is basically wrapped around this ball-like structure. So this is a ball-like structure and our DNA is roped around this ball-like structure. And there are, that 1.75 ton of DNA. So there are 1.75 ton of DNA. 1 ton is complete and 1 ton is 3 by 4th. So this is our octamer, and this octamer is basically present in this way. And this structure here is what we call H1 protein. So this structure here is H1 protein. There will be another octamer. There will be another octamer here. The DNA which is present between these two octamer, the DNA which is basically present between these two octamer is what we called a linker DNA. So it will complete similarly 2 turns and then there will be histone protein. So this DNA here is what we called a linker DNA. Linker DNA. So if we talk about this length is 10 nanometer. The height of the octamer is around 6 nanometer. So if we will see a DNA under microscope, we will see that this DNA is basically folded around a ball-like structure, which is basically histone. So this is fold-like structure. This is ball-like structure, That means histone protein. So if we see from the top view, this fiber would be basically typically a 10 nanometer fiber. This fiber would be a 10 nanometer fiber. This 10 nanometer fiber is basically called, this 10 nanometer fiber is basically what we call nucleosome fiber. Because it is made up of nucleosome, therefore it is called as nucleosome fiber. This nucleosome is then converted into another structure which is called as solenoid. Because of the binding of the histone protein, this is the DNA, and because of the binding of the histone protein on this DNA, this histone protein causes a turn. This histone protein causes a turn of 60 degree. This histone protein basically causes a turn of 60 degree. So gradually this histone protein causes a turn, and this DNA would be present in this form, and this model is what we called solenoid. So this is 10 nanometer nucleosome. This is 10 nanometer nucleosome, and this 10 nanometer nucleosome is converted into another structure which is basically 30 nanometer white, and this structure is basically what we called solenoid. Probably the major, this is the major kind of this solenoid. This solenoid is probably the major type of chromatin. This is the major type of chromatin fiber. So this is basically, or it is also known as chromatin fiber. It is also known as chromatin fiber. So this chromatin fiber is basically the major type of the chromatin material in nucleus during your interphase stage. And these forms, that these nucleosome and solenoids, are interconvertible into each other. That means this 10 nanometer fiber can be converted into 30 nanometer fiber, and this 30 nanometer fiber can be converted into a 10 nanometer fiber. So if we talk about this conversion, that is 10 nanometer fiber to 30 nanometer fiber, if there is high ionic strength, this 10 nanometer fiber is converted into 30 nanometer fiber. If there is low ionic strength, this 30 nanometer fiber is again back converted into 10 nanometer fiber. And this is the major form of the chromatin present here. If we talk about this chromatin fiber, these chromatin fibers can be of two types. These chromatin fibers can be of two types. The first kind of chromatin fiber here could be a euchromatin fiber. Or it could be a heterochromatin fiber. Euchromatin means this is the lightly stained part of the chromosome. This is lightly stained part of the chromosome, and this is darkly stained part of the chromosome. So if we talk about euchromatin and heterochromatin, these are basically the whole part of a chromosome. So if we talk about a chromosome, there is one chromosome. If there is one chromosome, there is one DNA. So this question can be asked, and this is asked in various examinations. So one chromosome means one DNA. So if we talk about this particular chromosome is basically a DNA molecule. On some part of the DNA there is heterochromatin, and some part of the DNA is euchromatin. What is meant by euchromatin and what is meant by heterochromatin? If you stain this part with a dye, if you stain this chromosome with the dye, there will be some lightly stained part and there will be some darkly stained part. The lightly stained part is what we called euchromatin, and darkly stained part is what we called heterochromatin. So if we talk about, let us say this is the part of the DNA, and this part of the DNA we are zooming out this part. So if this is the part of the DNA, let us say there is euchromatin. That euchromatin means that DNA is less folded. And if we talk about heterochromatin, let us say this is this heterochromatin part. So the heterochromatin part is basically more folded. So this is basically the heterochromatin part. So if you stain this part with a dye, this will be darkly stained because there is intense folding going out there. So this part of the DNA is basically lightly stained and lightly packed or loosely packed. So this part of the DNA is basically loosely packed. Whereas heterochromatin is this part. And heterochromatin a part is tightly packed, and heterochromatin part is always tightly packed part. So if we talked about the gene expression, a gene, if you have to transcribe this part, this can be easily transcribed. because the unfolding of this part is easy, because this can be unfolded easily. If you have to unfold this DNA, you can easily unfold this. So this can undergo easy transcription, easy translation. So if you are going to transcribe a part or unfold a part or translate a part, you are going to deal with euchromatin part. You are not going to deal with the heterochromatin part. Or you can convert this heterochromatin into euchromatin and euchromatin into heterochromatin. So this is the unfolded part. And this is the part which undergoes gene expression. So this part is basically responsible for gene expression. And this is unfolded part. This is tightly packaged part. That means this is tightly packaged part. And this tightly packaged part, there is no gene expression. There is no gene expression. If there is no gene expression, then means there is no transcription activity. So this is transcriptionally inactive. And this is transcriptionally active. So if we talk about the euchromatin part and heterochromatin part, euchromatin part is one which is transcriptionally active and heterochromatin part is one which is transcriptionally inactive. These parts are basically interconvertible into each other, and this is the solenoid model. This solenoid model, so this is the packaging of, this is the concept of packaging in eukaryotes. So we have already done this part. We have done this part, and this is the solenoid model. So this is the DNA. So we started from here. This is the double helical structure of the DNA, which is called 2 nanometer fiber. This 2 nanometer fiber curve around the histone protein. So this is the histone protein, that is the octamer new body, and this histone body is basically surrounds the DNA around itself, and there is 1.75 ton of DNA. So there is 1.75 tons of DNA around this histone protein. This nucleosome have H1 protein in between this part, and this kind of DNA is called linker DNA. This fiber here is basically what we called 10 nanometer fiber. So this fiber here is basically called 10 nanometer fiber. When histone protein is bounded to DNA, the histone protein bound होते हैं to this entire DNA, this causes 60-degree turn. So this causes 60-degree turn. 60 degrees, 60 degrees, 60 degrees. So in a complete circle, there are how many proteins of how many octamers? 6. So there are 6 histone proteins. So if we look from the top, this would be looking like this model, and this part is here. So there is a 60-degree turn. There is a 60-degree turn due to binding of this histone protein, and this is what we call solenoid model. Further, this solenoid model is then converted into super solenoid structure. This super solenoid structure is 300 nanometer fiber. So this is converted into a 300 nanometer fiber, and this structure is what we call super solenoid. This super solenoid is then packaged into a higher level of packaging, and this higher level of packaging is what we call chromosome. So this 300 nanometer fiber is converted into chromatid, into structure what we call chromatid. So this chromatid structure is basically 700 nanometer fiber. This 700 nanometer fiber, when this chromosome replicate, so one arm is 700 nanometer, so the complete double structure of this chromosome is 1400 nanometer. This is the highest level of packaging present for DNA. That means these chromosomes or rod-like structure are basically highly folded structure, intensely folded structure of a DNA molecule. So this is all about the levels of DNA, levels of the packaging of DNA. So if we talk about, if we have a quick revision, we have this DNA. This DNA here is a 2 nanometer fiber. This 2 nanometer fiber is then converted into 10 nanometer fiber. 2 nanometer fiber is converted into 10 nanometer fiber due to binding of histone proteins. So this is histone assisted binding. Away from this is converted into solenoid structure. Solenoid is 30 nanometer structure, and this is due to bands which is caused due to the binding of histone protein. So this is converted into solenoid. Further, this solenoid is converted into this 30 nanometer fiber is converted into 300 nanometer fiber, and this 300 nanometer fiber is what we called super solenoid fiber. Then there are other proteins for folding the chromosomes, and this is converted into an chromatid arm, and this 300 nanometer fiber is basically converted into 700 nanometer fiber, and 700 nanometer fiber is basically converted into 1400 nanometer fiber. So this is all about the DNA packing mechanism. I hope you liked the video. If you have any doubts regarding the video, please let me know in the comment section. I will come out and reach to every one of you and try to give answers. Thanks for watching video, and don't forget to hit like and subscribe button. See you soon in the next video.